Hi, my name is Lawrence. I am a CF student studying Honors Health Sciences at miRNA College, and today I'm excited to present my project, miRNA Discovery for COVID-19. For my project, I aim to identify a therapy against COVID-19 that is both widely accessible and highly effective, something called RNA interference, which is caused by microRNAs. To identify such microRNAs, I programmed a computer model that takes a multi-layered approach to pinpoint genetic sequences that could potentially bind to SARS-CoV-2's genetic material, silence it, and stop its replication. The idea of RNA interference initially sprung from another idea I was pursuing. I just wanted to learn more about how the human genome works, since only about 5% of the genome is expressed in proteins. The rest, the other 95%, plays a largely mysterious role. We do know that a series of sequences called microRNAs, or miRNAs for short, however, are engaged in silencing genes by binding onto them. Although I was initially interested in brainstorming ways to apply this RNA interference phenomenon to cancer research, the COVID-19 pandemic hit, and then, I wondered, how could RNA interference silence the replication cycle of SARS-CoV-2? It was a challenge to determine a method that could accurately predict such sequences and such binding locations. But after doing a bit of research, I came across miRDB, a tool developed by researchers at the University of Washington that could actually predict, or accurately predict, in some ways, the potential binding sites of microRNAs. Using miRDB, I wrote a computer model that would automatically upload SARS-CoV-2's genetic sequence and then save the results in an automated fashion. I further filtered the return candidates by having my computer program cross-reference them with existing scholarly research on PubMed and also pinpointing binding sites that can occur in conserved regions, which basically are regions that are of interest. In the end, I identified 169 microRNAs that could be potential candidates, though one really stood out, a candidate called miR302C. miR302C was ranked 10th of 169, but it's not only its high ranking rather, that makes it striking. In fact, prior research discovered that if you overexpressed miR302C in mice infected with bacterial pneumonia, the miRNA can actually improve survival rates. This makes it a very promising candidate for RNA interference treatment against COVID-19. As a future step, I'm interested in testing in vitro how overexpressed miR302C in lung cells can curb COVID-19 replication, as well as apply my computer program to identify miRNA from emerging variants. For this project, I received the guidance and support of Professor Angela Keene, science fair teacher at miRNA College. I really enjoyed working on this project, but more importantly, understanding how microRNAs work and how they can potentially inhibit genetic sequences, and figuring out, which microRNAs are involved in these sequences. I really look forward to meeting you and to answering your questions that you might have. Thanks for tuning in and have a great day.